Opening our conference today is the Professor of Medicine at Michigan State University. He is also the editor of the Journal of Anti-Aging Medicine and has dedicated many years to studying progeria and related accelerated aging syndromes. He is convinced that the evidence from these diseases, together with the fact that germ cells and cancer cells do not age, indicates that aging is a regulated process, that is, a function of gene expression, and not a function of wear and tear. Talking this morning on the subject of cancer, would you please give a warm welcome to Dr. Michael Fossel.
The first question, it is. I was going to say, is the lavalier going? And it is. Thank you very much for being here this morning. I brought a little gift. I brought my garden. It actually is my garden. It's a nice place to be and not to age, maybe. A lot of you probably wonder why we're talking about aging and cancer together. It's sort of an odd conjunction in a way. And I think that you hit it right on the head when you said that part of the issue is that cancer cells don't age when we wish they would, and the rest of our bodies age when we wish they wouldn't. But we're going to focus this morning on cancer, and then tomorrow I'll talk to you more about aging itself. The first question you might ask is, what is cancer in the first place? And an easy answer to that would be this. It's a disease of inappropriate cell division. Now, our body cells divide all the time, but they divide under a closely regulated set of mechanisms, both within the cell and between cells. The problem in cancer cells is that those regulatory mechanisms fail, and the cells continue to divide when it's totally inappropriate. Most normal cells, as Len Hayflick pointed out back in the 60s, slowly senesce. In fact, they do that with a loss of telomeres that was shown nicely in the 1990s and continues to be shown. The problem with cancer cells is that they in fact don't do this. There's some initial shortening of telomeres, but then they maintain them, and they in fact fail to age. And we wish they would. The question, though, that comes up to me as a clinician is not so much what is cancer, which helps us to answer this question, but the major question itself, which is what can we do about it? The facetious answer to that is it's very easy to kill cancer cells. I like to say that I can kill anybody's cancer cell in this room within 5 minutes, no exception, 100% guarantee. There's a little drug called potassium cyanide. Very, very effective. There are some side effects. It's lethal with the rest of you, too. But when you think about it, that's precisely the problem that all of us have with treating cancer. You know, the worldwide, in developed nations, the worldwide survival rate for average cancer now is about 50%. Some a lot worse, some a lot better. The problem, though, is not getting killed in the process. And what we've long sought is sort of the holy grail for cancer, the magic bullet, something that you can pharmacologically fire at the body, hitting only cancer cells and not hitting normal cells. And it's obviously not been easy. In fact, historically, the feeling has been it was impossible. There are a set of intrinsic defenses in the body against cancer. I'm going to divide them into extracellular ones that I'm not really going to talk about this morning. And there are three sets. Cell-mediated immunity, where your body recognizes cancer cells, hopefully better than we do pharmacologically, and kills them. And in fact, for most cancers, it's very effective. Everybody in this room probably has cancer cells that you will never see. Your immune system will kill them off very nicely, although it fails to do so as you get older. The second is enforced apoptosis, or stachycardiasis, we'll talk about tomorrow. And the third, well, I should say it's kind of intriguing in that this is, in another sense, almost an immune phenomenon. Your body's neighboring cells are telling a cell to commit suicide. Again, the problem in cancer is that it ignores those signals. The third applies only to bulk tumors, to solid tumors, and that is required angiogenesis. If a malignancy cannot induce angiogenesis, it fails. For example, a breast tumor, an ovarian tumor will fail. It doesn't apply to leukemic tumors and other small cell tumors like that. But there are a whole set of intracellular defenses that most of us don't pay much attention to, and that turn out to be critical in what's coming to be some changes in our ability to treat cancer. And these are the intracellular defenses. The first of which is DNA sequestration, or hiding, and repair. The eukaryotic cells, you for example, are the only cells that effectively hide the DNA one place and all the mitochondria hide all the bad things someplace else. So your DNA is protected from the most dangerous place in your cell, the mitochondria. The problem with that is that sometimes the sequestration fails, and your body tries to repair the damage and fails that as well. DNA molecules are the only molecule in your body that's repaired. All the others you simply turn over. You throw them out, you recycle, you build new ones. The DNA, you can't afford to do that, and you repair it. But again, that fails. The second mechanism is the cell cycle breaking system, which we've known about now for about 13 years, more or less. And we'll come back to it. The third is cell senescence, that we'll focus on a bit this morning in a minute. Some people would compare these three to a sort of a failure of regulatory mechanisms within the cell, sort of like the accelerator of a car. And it gets stuck down, these cells cycle through, they try to produce more cells, and the brakes fail, and cell senescence is sometimes compared to the gas tank that should run dry but doesn't in malignant cells. If we go back to the 1980s, historically, the feeling was that all cancers were a problem with DNA damage. And here what happened was that normal cells accrued damage to the DNA and failed to repair it. And the problem with that was that the damage was felt to be random. Could be any one of 100,000 genes in effect, or at least in those days felt to be. That each of these was unique, and that there were countless possibilities. In other words, let me put it more graphically. If I went back to the 1980s and I talked to a cancer researcher in public with a microphone, and I'd say, can we cure cancer? The answer would be, if you put enough money in my grant, yes. But privately, over a glass of wine in a hot tub, the answer was never. And the reason they'd say was something ironic. They'd say, there's no such thing as cancer. I say, what do you mean by that? And they'd say, well, there's her cancer and his cancer and my cancer and your cancer, and they're all different. There is no generic cancer. There's no such disease, they'd say, because every single one is unique. That's very frustrating because it implied that there were so many targets we couldn't possibly hit them. We might be able to work on this childhood leukemia, but not that breast cancer, this ovarian cell cancer, but not that lung cancer. So it was a very pessimistic view. And as it turned out, unrealistic too. Because in about 1989, we began to understand something of how the cell cycle breaking system worked and the implications it had for cancer. Essentially, what your cells do, nature's been at this for 3.5 billion years, and it knows a lot more than we do, certainly more than I do. Not only do you repair damage, but your cell is continually fingering the chromosomes, looking for damage. And when it finds it, you get the release of damaged DNA binding proteins that trigger the main molecule. The big player there is that P53, became the molecule of the year back in about 91 in Science, and a host of about 12.5 other molecules that together compose the cell cycle breaking system and prevent the cell from continuing to divide. Again, just like a break. So now by 1993, the model had changed. The idea was that every cancer has to have two problems, DNA damage and failure of the breaks. And so now the pessimism had changed. And you found people saying something they'd never said before, even very conservative approaches. For example, again in Science. They were saying that there is now a distinguishing fundamental characteristic difference between cancer cells and normal cells. The implication of that was that we could use that as a therapeutic leverage to go after cancer cells and not harm normal cells. You see the surfacing again of this magic bullet thought. Because now you're looking at only 12.5 targets. They're fairly specific and fairly well known. You could target them, and therefore the therapeutic targets were promising. For example, P53, 60% of colon cancers. Nice thought. You could go after them fairly easily. As it turns out, that wasn't all that the cell does to defend itself against cancer. And that has to do with the telomere. As it turns out, most cells simply run out of fuel. Effectively, even if the brakes fail, after a while the cell runs out of gas and stops dividing, and in fact undergoes wholesale changes in gene expression. So the normal cell then loses telomeres and becomes senescent. What cancer cells do is they end up re-expressing a molecule, telomerase, and resetting the telomere. Just as an aside, I should mention that there's a sort of a flippant disagreement worldwide about how to pronounce that. There is one minor and two major ways there are of pronouncing the word telomere. Most Americans will say telomere. Most of the British and the Canadians will say telomere. I noticed that they never say telescope, but if they want to do that, they could call and argue with me on the telephone, I suppose. These are the same people, by the way, who say telomerase, even though they say telomere. The third pronunciation is located only in Japan. I don't say this facetiously, it's telomere, and the Japanese is teramarasu for telomerase. In fact, when my book came out, the Japanese editor sent me a letter in which he spelled it teramere. I thought he was joking, but he was serious. We corrected the proofs. So by 1995, we were just beginning to have a third model, or an extension of the model. We now knew that every cancer cell essentially had to have the DNA damage, had to have bad breaks, and furthermore, theoretically at least, had to express telomerase. The exceptions to this turn out to be sort of interesting, and it may not matter practically. So the feeling was, at least simplistically, that telomerase was a reliably defining molecule for cancer, and therefore there was a single target therapeutically. If that was true, you could see how that would be exciting, because it would be the first time that we could aim at cancer and hit it without offending normal cells nearby. So the three steps then went something like this. You started with normal cells that underwent any number of thousands, tens of thousands of possible different sorts of damage, and then had to have maybe 12.5 different damage components of the breaking system, and then had to singly express a molecule H-TERT, the enzymatic component of telomerase. And that's enough to have cancer. So again, that final target is one that intrigued a lot of people. And as it turns out, with a few exceptions, it's present in all cancer cells, and I'll come back to that. And it's not found in normal cells. That's not true either, but I'll come back to that. The first report of this came out in '94 in Proceedings National Academy, and these are two early reviews: Chris Counter's work, and then Jerry Shea and Woody Wright, and Cal Harley's work. More importantly, that implied you could actually inhibit telomerase, and if you could do that, you could kill cancer. And in fact, that works very nicely. What Feng did in her original article was she took normal ovarian cancer cells, are they normal? Cancer cells, ovarian cancer cells, and normal ovarian epithelial cells, put them essentially in the same petri dish, and added a telomerase inhibitor, in this case an antisense molecule, and showed that she could reliably kill cancer cells and have no noticeable effect on the function and longevity of normal ovarian cells. Bingo, magic bullet. As you might guess, there are problems with that. But what it did was inspire a huge amount of research on what now is literally thousands of different molecules that act as telomerase inhibitors. Curiously, some of these telomerase inhibitors turn out to be known oncogenic molecules, and some known oncogenic molecules turn out to, strangely enough, inhibit telomerase. So what we find is that cancer cells, as they go along, continuing to divide, can be stopped very effectively. When we put in telomerase inhibitors, it effectively enforces telomerase. It makes the gas tank run out, even though the accelerator's pushed down, even though the brakes have failed, those cells actually come to a stop. So where are we with regard to telomerase? They're effective, entirely effective, remarkably effective. They are specific, again, with a few interesting exceptions I'll come back to in a moment. The problem is, are they safe and deliverable? Okay, it's not enough, for example, to have a molecule that effectively kills only cancer cells, ignores normal cells, but, say, interferes with cardiac conduction for 5 minutes, or causes an overwhelming inflammatory response. You see what I mean? It's nice in distinguishing cells, but in terms of a multicellular organism, that would be a failure. Likewise, it doesn't do if you have an RNA antisense molecule that you can inject, but in your blood it has a half-life of 5 minutes. It's just not effective if it never gets into the cells to do the job, because strangely enough, people aren't petri dishes. So here are some of the caveats. First of all, there are certain numbers of stem cell lines that are telomerase positive. That turns out to be very good, because if not, you'd run out of blood cells early in your life. An example here is the hematopoietic stem cell line in bone marrow. The second example would be, in this case, GI villi. The deep crypts are telomerase positive cells to continue to regenerate your GI lining. Third example would be hair or skin cells here. I don't have the hair follicle, do I? But normal skin cells throughout the dermis, basal dermis cells. And in fact, hair follicles are another example. There are about a half a dozen known telomerase-positive stem cell lines in human beings. Most of the time, these don't continually express telomerase. They express telomerase in response to intracellular signals. They turn it on, they turn it off. It's under good control. What happens also, though, second problem, is that there are some cancer cell lines that are telomerase negative. But that turns out to be useful. It turns out that all known malignancies and all known metastatic cancers are telomerase positive, again with some very minimal exceptions, but they are minimal, and that the telomerase status of a tumor seems to define whether it's benign or malignant. Example, childhood neuroblastomas. We've known for a long time there's some benign, some malignant, and we had no way of telling whether your child's was benign or malignant except waiting. But the benign ones are telomerase negative, and the positive ones are the malignant ones. Interesting. The second problem is that if we have a telomerase inhibitor, this will have a slow oncocytal activity. You know, as opposed to the potassium cyanide that works in seconds to minutes, most of the oncotherapeutic molecules we currently use work in a matter of hours and days. We're dealing with molecules now that work on the order of weeks to months, which brings up a real problem, which is, are you going to die before it's effective? Generally, that's not the issue. And in fact, what's interesting is that these molecules are far more effective on the more aggressive tumors that are rapidly dividing and therefore rapidly senescing. So the nice thing is that the indolent tumors are the ones that it tends to work slowly on, but the rapidly dividing cells, it works on very fast anyway. A fourth problem is critical locations. You know, most of us regard lipomas as benign, and they're benign histologically. But if I put one in the middle of your medulla, in your respiratory center, it's malignant anatomically. So you can have a perfectly benign tumor histologically that kills you anyway. It doesn't help you that it's benign. Nor will telomerase inhibitors help one bit. Wishful thinking may help. The fifth problem is long telomere cancers. Now I should put that differently. Long telomere malignant cells. We know a couple of lines in the laboratory that have long telomeres. We don't know of any cell within the body, in vivo, a malignancy within the body that has long telomeres, but I think it would surprise most of us if there weren't a couple. The most pressing issue, though, is not these five. It's the finding practical telomerase inhibitors. Let me point out one more thing about these stem cell lines. I forgot to mention germ cells. Now, a classical problem with any of these are the same problems we have with current cancer therapies. All of these telomerase positive lines tend to be associated with side effects in malignancy therapy. For example, GI side effects, immune side effects, hair loss. These are the sort of things we currently see with a lot of chemotherapeutic approaches. But germ cell lines also have this extension, at least, for example, the cells surrounding ova and the germ cell lines within the testicle. Practically speaking, though, the issue has to be compared to the risk you have anyway. I mean, if you tell me that I can be alive but infertile, or dead but fertile, which is not a difficult decision. So let's go back to the practical issue, finding practical telomerase inhibitors. Where are we now with this? And the answer is, most of these, a whole host of these actually, have passed through in vitro trials. We probably have in the order of close on 100 telomerase inhibitory molecules that work in vitro. They have been effective in animal studies, and there we're looking at in the order of a dozen or so different sorts of molecules that bind in different ways that function in animals. And there are now a handful of phase 1 trials in the US working in vivo in human patients now underway. So far, so good. I think we could probably expect to see phase 2 or 3 trials beginning in the next couple of years. But still, that's a ways away, and it doesn't help you today. I want to thank Phil Mickham, Brad Eitner, and the rest of the conference, namely you people, for having me here at all. And we have actually a few more minutes. I want to also point out that Marios Kariazis is going to be talking tomorrow on apoptosis. But before going into questions, let me say that since we have a couple of minutes, what we can do, a lot of you had at least sort of indirectly have expressed an interest in the progeric children. And if we have a couple of minutes here, I'll throw on a couple of slides of those progeric children and talk about that disease for about 5 minutes. Questions, though, first on cancer and intracellular mechanisms. Any?
Anybody has a question? Would they like to? The gentleman here, one question only, please.
Good morning. I wondered if you might be able to describe something called polymerase chain reaction blood testing.
I couldn't hear that.
Reverse polymerase chain reaction blood testing.
Reverse
Polymerase chain reaction blood tests.
You know, generically, polymerase chain reaction is not something I really deal with or know about. It's certainly like in any of these approaches. I mean, certainly not just telomerase is a useful therapeutic or a useful tool in the laboratory, but I'm not sure specifically what you're asking about it. You know, we've been using it to identify telomerase, for example. You're right. You know, the classic example here has been to find telomerase expression in urine to look at genitourinary tumors. There are a lot of problems with that. If you really want a lot of details, there's a textbook I've been writing for Oxford University Press, and there's a huge chapter on cancer. There are some 4,000 references in the thing. It's not light reading. I promised Oxford University Press I'd have it written in 6 months. That was 2 years ago. I'm getting there. But I'll be happy to give you a copy. I've got it with me.
Okay. Sorry, Michael. If anyone's got a question while Dr. Fossel is answering, please put your hand up so I can find you. The lady here was first, sir. Sorry. Sorry, you do need the microphone.
You gave the example of the pediatric neuroblastomas and differentiating benign and malignant via the presence of telomerase. Mm-hmm. Is there a clinically available means of measuring telomerase? And as a side question, is there a telomerase level above which you might assume there is a malignancy other than that produced by regenerative tissue?
Okay, everybody I assume heard that. There are actually 3 things you can measure, and they have had different utilities. One is telomere length, which, just giving a generic answer, is useless. And I could explain why, but basically it doesn't have a clinical utility in most human cancers. The second is to look at telomerase expression, that is the actual presence of telomerase within cancer normal cells. And as it turns out, that's somewhat effective and somewhat useful, but certainly not reliable. The third thing to look at is telomerase activity. As you can find cells that express telomerase, but for one reason or another have no telomerase activity, no measurable telomerase activity. So while it's there, it appears to be blocked or at least not functional. On the other hand, you can sometimes find cells where there's no measurable telomerase, and yet you can show telomerase activity, which is a real eye-opener. And there are some interesting explanations for that. But basically what you want to measure is telomerase activity. And overall, the reliability is about 92%. That is, 92% of all human malignancies, taking them all, you know, whether they're metastatic or not, roughly speaking, are telomerase positive. And luckily, again, the more malignant ones tend to be more positive, and the later stages tend to be more positive. But again, not uniformly. Let me remind you of a historical analogy here. If we go back to 1873, there were still, most scientists, medical scientists worldwide, in what served as developed nations in those days, would argue that microbes had no role to play in human disease. I've got some great quotes, as opposed to Pasteur and Koch, who knew better. Now, most of us these days feel strongly that cellulitis that we see clinically is caused by bacterial disease. And the reason we think that, besides just personal bias and superstition, in effect, is because when you treat human cellulitis with antimicrobial agents, for example, your standard antibiotics, they respond very nicely. But if you try testing for the presence of bacteria, in the best of samples, somewhere between 25 and 33% of cellulitides can be shown to have microbial growth. So does that mean microbes don't cause human disease? What I'm suggesting is that sometimes our ways of measuring, in effect, telomerase activity too, may not be sufficiently sensitive, but it still may be that a telomerase inhibitor may be effective. I'm not saying it's true, I'm saying it could be true, based on that same sort of analogy. We'll just have to see how it plays out.
Good morning, Dr. Cohen. Thank you for a very interesting lecture. Dan Stein, MD, from Tampa, Florida. Several things. Number one, I think we will see a recurrent theme throughout this meeting about our ways of measuring. And I think that the way the lens that we look through and the accuracy and clarity of that lens skews our perception of all objective data. You've addressed cell reproduction as a characteristic of malignancy, and the growth restraint is one characteristic of normal cells as part of senescence. What about the ability of cells to invade adjacent tissue and compete with and effectively surpass those cells for nutrients, oxygens, etc.? And secondly, what about the ability for metastasis, without which most malignancies would not be life-threatening?
Well, with regard to metastasis first, again, there are a number of cancers that can be fatal without metastasis, but you're right, you know, a great many of them do. The major characteristic, again, put flippantly, the major characteristic of cancer cells is that they're biological sociopaths. There's no restriction on what they're willing to do. These are the people who have old cars in their front yard and you know, beat the neighborhood children up. But when you think about it, in a more serious vein, that's selected for within cells. If you have a benign tumor that doesn't continue to divide and invade and is not willing to metastasize, the neighboring cells won't have usually too much problem with that. That is, you've got sort of a passive tumor. And my guess is it may even be that those are the tumors that your immune system finds a little easier to localize too. I don't know that. But selection pressure on malignant cells favors cells that are aggressive. Those are the ones that you see are the ones that will tend to be aggressive. If you don't see them, they were passive. So again, back to your point about measurement, you know, there's an inherent bias in what we see clinically. We only see the worst of the worst. It's like me in the hospital. I remember being a medical student back at Stanford. We were a tertiary care center. I used to think that the only people who ever got pneumonia were immunocompromised people on multiple chemotherapy who had cancer. The normal people didn't get it. And the reason was, that's all I saw who had pneumonia. Well, there was a bias there. And it's not until you go out into the neighborhood you realize that everybody gets pneumonia. I was only seeing the odd cases. And there's a lot of that. And again, naturally, we pick the aggressive tumors here.
A question from Dr. Pierpoli.
Michael, an experimentalist questions. I'm not in the field, just a curiosity. When you paint the skin of a mouse with a carcinogen, yeah, to induce cancer, you know, it's a very common procedure to induce cancer in rodents. Do you see a progressive alteration of telomeres? And how far, if you stop painting the carcinogen, you can see a possible regression to normality? There is a repair mechanism in the healthy body which, in spite of carcinogen, would allow, let us say, a regression of the induced lesion at the telomere level.
You're exactly right. You know, the body has not an infinite, but a remarkable capacity to deal with damage and insults. And that's really the hallmark of biological organisms as opposed to physical structures. You know, I wish my car would regenerate, but it doesn't. But most of us as biological organisms put up with a great deal of insult, and in fact, you know, again, thinking of Nietzsche, what doesn't kill us. And it's true biologically, though. You know, the whole issue of, what is it called? Oh, who's the Dane who's into this? But, you know, we tend to be stronger because of it. So we do repair DNA damage. The telomeres in those cells have there are two sorts of reasons the telomeres in those cells shorten. One is that they continue to divide, but the other is you can actually show that there are probably direct mechanisms that cause telomere loss related to reactive oxygen species, and probably some other things. I mean, there have been some links with heavy metals too. There's a remarkable amount that we don't know about that, obviously. It's very confusing literature. I can't help you much. Let me stop there. I'll just show you a couple of pictures of the progeric children anyway, and we'll move on. These are children without childhoods. This came out of a little conference I was giving in Marrakesh, hence the Arabic. There, let me go back to that. That's Rachel. Rachel was 7 years old when that picture was taken. She has no hair, very thin skin. You can see the beak-like nose. Cute little girl. We actually checked her telomerase length, her telomerase expression and telomere length, comparing it to my daughter's when my daughter was the same age. You'd be surprised what my daughter volunteered for the right inducement. And she has normal telomerase expression in her lymphocytes, which doesn't surprise us because these children have normal immune function. It's not an old immune system they suffer from. It's old skin and old vessels and old joints. There's a 5-year-old, 19-year-old boy, one of the oldest, 8-year-old. This girl, strangely enough, is Vietnamese. What's interesting about these children is they look much more like each other than they look like their families. She doesn't look Oriental to anybody. And in fact, the only difference we see in these children is that children with dark skin tend to look like their parents, but everybody else, facial features, they all uniformly look like each other. 5-year-old, same thing. This was first described. Currently, we have about that number, about 4 dozen. I'll tell you why, too. This is what usually happens. We usually pick them up shortly after birth, first year or two, failure to thrive. The average age of death is 12.5 years. These children die of heart disease and strokes. Curiously, they don't have high cholesterol, they don't have high blood pressure, they don't have high glucose, and none of them smoke. And does that mean that the standard model of atherosclerosis is wrong? No, it doesn't mean that. It just means that there's some things we don't know about with regard to that model. Here's where we see it. We tend to see a correlation with countries that have good healthcare systems. And probably, again, speaking of bias, that's why. You know, if you end up with this in Chad or in Bangladesh, you die young for other reasons, failure to thrive, you don't have food anyway. If you're lucky enough that you survive that, your parents can't afford to take you to a doctor who can't recognize it, who, if he did, wouldn't know who to call anyway. So we tend to see these children, for example, in Europe, North America, Argentina, places like that, that have fairly good healthcare systems, Mexico, but we don't tend to see them elsewhere. That Vietnamese girl was sort of an exception in a way. The pathology on these children, they tend to have bad skin, bad bones, bad vessels, but curiously enough, no change in the brain, none in the immune system, and no increased risk of cancer. These children are awfully bright, they just look old, and they die of old age diseases with regard to the vessels. Terrible arthritis. Affected cells, not surprisingly. The keratinocytes, fibroblasts within the skin and within vessels, endothelial cells within vessels. And again, not surprisingly, the unaffected cells are neurons, myocytes, leukocytes, and so on, matching what we see clinically. Pathology, something like this. We see a cell senescence deficit in the affected cells. Some people think it's an epigenetic mosaic. I do. In short, what I'm thinking is going on has more to do with the way that genes are expressed and the actual change in gene. And then it's about time to move on, though. It's not this. Usually made clinically. You can test for hyaluronic acid, telomere length, and fibroblasts, and you find advanced cell senescence. Questions about those kids? I hope not, because it's time to move on. Thanks very much.